Oh, good morning again. Let's see, it is Sunday. So for me, this won't be up till Monday. So you'll be taking the exam today, Monday. Again, I sent out some instructions on an announcement, but basically it'll open up Monday at 7 in the morning. It'll be open for a week, 6 days till Saturday. Once you start, you have about 55 minutes. It's 50 multiple choice, 3 short answers. They're pretty wimpy short answers. Shouldn't be an issue. Short answers have to be manually graded. So as they come in, I'll start grading them and post the final grades. Let's see. So we're going to continue with the respiratory system. Next exam will probably be respiratory, maybe respiratory digestive. Might make 2 smaller ones. That seems to be easier to do. But let's continue on with the respiratory system. Now we've gone through the external areas, through the nasal cavities, pharynx, and done with the larynx right here. So what I'm going to do, we'll go through the rest of the airways today. I'll do a few stick pictures here, then I'll go to the small screen and we'll go through some real pictures that are a little easier to see. Basically, after the larynx, you have the trachea. Now, if you look at the trachea, I'll show you some pictures in a bit. You've seen it on the models a little bit. But the trachea is a little unusual for a tube that you find in the body. Most of your tubes, the esophagus, the aorta, the intestines, the ureters, the urethra, almost all of your tubes are muscular. They're just soft-walled tubes with some smooth muscle in them that can, contract and reduce their diameter, move things along, that sort of thing. Trachea is a little different. There's not a substantial amount of smooth muscle in the trachea. If you look at it, it's got these heavy sort of C-shaped hyaline cartilage rings. So if you looked at a cross section, you'd see these hyaline cartilage rings kind of all stacked up. Kind of looks a little bit like a slinky. Not a lot of muscle. In fact, just a little insignificant bit of muscle here and there towards the dorsal end. So not a lot of muscle. Now, why is that? Well, it's because of the way we breathe. Mammals use what's called a negative pressure respiratory system. If you imagine this is your trachea, I'm just going to draw kind of a little tube like this. There, it's just a tube. The pressure out here in the atmosphere, let's just say it's sea level pressure, 760 millimeters of mercury pressure. It's on the outside. Now, when we inhale, you did a little lab on the mechanics of breathing. I may go over it a little bit, but basically the diaphragm, it's a dome-shaped sheet of muscle. I think you've already seen it in the lab, but it's up like this. When it contracts, it pushes down. What it does, it increases the volume of the chest. Now, because of Boyle's law, if you increase the volume on a unit of gas, you're going to drop the pressure. So when the diaphragm contracts, the pressure down here in the lungs and in the airways, it gets lower. It might be, 756 millimeters of mercury. So pressure in the tube, in the airways, in the lungs is lower than the outside pressure. What that's going to do, it's a force. You have more pressure outside than inside. That's going to tend to collapse the wall. So if you tried to use a straw. Think of a straw as an example. If you cut a piece of the esophagus off, kind of gross, but stick one end of the esophagus in your coffee, other end in your mouth, and try and suck coffee through an esophagus. It won't work, because when you create that vacuum or lower pressure inside the esophagus, the atmospheric pressure just squashes the walls closed. Of course, you squash the walls, resistance gets too high, it's impossible to suck fluid through it. Any tube would do that. You can't use an aorta for a straw, can't use an esophagus, can't use the intestines, clean them out first. But if you took a trachea, stuck in your coffee cup, and tried to suck off through it, hey, your trachea would work as an emergency straw. It's disgusting, but it would work. That's because those C-shaped hyaline cartilage rings, they give it support. They prevent the trachea, and other airways to some degree, from collapsing when you inhale. That's why if you got punched in the neck and you break that cartilage in the larynx or trachea, you'll suffocate. When you try and inhale, that collapsed cartilage no longer supports the pressure. Every time you inhale, the walls of that thing collapse up and you can't breathe. So, trachea, it's mainly a conduit. Basically, if you look at it, it looks a bit like a vacuum cleaner hose. For the same reason, mechanically, we mimic this with our vacuum cleaner hoses. They'll have those little supporting or reinforcing rings around it, because it's got the same problem. When you turn on a vacuum, the pressure's lower in the hose, hence the name vacuum, than it is outside. That tube would collapse. So the trach is a lot like a vacuum cleaner tube or hose. It's basically just a conduit. It brings the air from kind of the upper respiratory tract down into the chest and eventually to the respiratory portion of your lungs. Now, a couple other things in the trachea. You looked last semester when you were looking at your histology. The epithelial alignment of trachea is columnar epithelia. That was that pseudostratified ciliated columnar epithelia you had, kind of off at a weird angle, so it looks stratified or a little bit stratified, but it's not. It's a simple epithelia. But it's also ciliated, and it had a lot of goblet cells. Again, the reason for that, you don't want a lot of junk in your lungs. The functional portion of your lungs is very delicate. Now, occasionally bits and pieces get in there. This is kind of your last-ditch effort to get that junk out. The goblet cells create mucus, which is going to be laying in a kind of sheet on top of those cilia. When the cilia beat, they beat in a coordinated manner, and they just take that mucus and they move it back up to your pharynx where you can swallow it or spit it out or whatever you want to do with your dirty mucus. It's the mucociliary escalator. That way when debris gets in here, bits of food, whatnot, the mucus is sticky, kind of like flypaper. Junk will stick to it, the cilia move it up, you can cop it out. So you do have ciliated cells in there as well. Now, what we're going to look at next, we're going to go from the trachea to the rest of the airways. And keep in mind, from the external areas all the way down to sort of the respiratory portion of the lungs, those alveoli, all of the tubing we're looking at, it's sometimes referred to as anatomical dead space. The reason for that is the walls of all these airways, they're very thick, they don't have much surface area, and they're very poorly perfused, not much of a blood supply. There is not any significant transfer of material between the air in these tubes or airways and the blood. So this is acting like just a conduit. In fact, the anatomical dead space on average in a human is about 150 milliliters. That means that first 150 milliliters you inhale, it's just filled up the tubes. It isn't even getting to the spot in the lungs or respiratory portion where you can get diffusion of oxygen and CO2 into and out of the blood. So we'll follow down the trachea. Now the problem is, you got one trachea, you got two lungs. You know, you might have a lung here. I'll draw some lovely lungs here and a lung over here somewhere. You got to split it in two. So there's kind of a hierarchy of branching. We'll start with one trachea, and it will split going to the right and the left sides. Okay, that's kind of terrible, but that's the idea. You got two branches. These are called the primary bronchi. So you have a right and a left primary bronchus. I'll just use a one primary bronchi. Primary bronchi, you got two, one going to the right, one going to the left. So the right and left primary bronchus, they go into the lung. Now, when we get to the lungs, human lungs are separated a lot of your internal organs, like the pancreas, like the testes. It's separated into lobes, subcompartments. Again, part of the reason for that is to compartmentalize damage, infection, parasites, that sort of thing. Now, when you look at a human lung, the right lung, this is the right side over here, has 1, 2, 3 lobes. I'll show you real pictures eventually. On the right side, the top part, that's the superior lobe. The one in the middle, surprisingly low, middle lobe. And the lower one is the inferior lobe of the right lung. The left lung is smaller. In humans, it's only got 2 lobes. Part of the reason it's smaller is that left ventricle. It's much bigger than the right. It kind of projects into the left side of the thoracic cavity a bit, takes up some of that space. So the left lung has this big depression in it. I think it's cardiac notch or depression. That's where the left ventricle is kind of taking up some of the space. So just 2 lobes on the right side. Superior, inferior, that's it. Now, once we get into the lungs, with these primary bronchi, they're going to split again. One branch goes up here to that first lobe, one branch here, and a third branch down to the third lobe. This second set of branching, creatively enough, these are the secondary bronchi. Let's draw now here, secondary bronchi. They go to each lobe of the lung. Now on the right lung, because you have 1, 2, 3 lobes, you have 1, 2, 3 secondary bronchi. They're sometimes also called lobar bronchi. Same thing, I'll use secondary. And I should mention these lobes, when you look at a fresh lung, you can get your fingers in there. I mean, they're big divots or fissures. They're not just a little groove or anything. There's a big separation here. So the right lung has 3 secondary bronchi. The left lung, there's only 2 lobes. So guess what? There's only 1, 2 secondary bronchi. So they're not quite symmetrical at this point. So those are secondary bronchi. Then if you go a little further, now this next subdivision is a little tougher to tell, but within each lobe, there is some connective tissue sheets, kind of compartmentalizing the lobe into segments or lobules. That's the next branching pattern. Again, these connective tissue sheets further self-compartmentalize the lobes a little bit, prevent the spread of damage, infection. But these smaller lobules are supplied by another airway, so it branches again. And 3 guesses what this one is called? These are tertiary bronchi, third order bronchi. And they all do that. So we got a lot, and we won't name them, there's just a bunch of them. But those are the tertiary bronchi. And they supply each of these lobules or segments in the lung. Now, this system of tubes will continue and go through many more generations of branching. We have trachea, primary, secondary, tertiary bronchi. Typically in labs or on cadavers, that's about as far as I go. But there are more branches. The name changes. There are much smaller branches coming off the tertiary bronchi, and those will be called bronchioles. So it's trachea, then you got the primary, secondary, tertiary bronchi, and the tertiary bronchioles split into bronchioles. There are many generations of bronchioles. They branch, get smaller and smaller, and feed into all the different parts of the lung. They get really small, like say, a millimeter in diameter, some of them pretty tiny. Most of them are not named. But that is the end of what we call the bronchial tree. I'll show you a picture of a casting in a bit. So you go from the external nares all the way to the end of this system, and the system of bronchioles will end. The smallest ones we'll look at are, they call them terminal. Bronchioles. Terminal bronchioles, terminal is the end. That's the end of the line for this sort of anatomical dead space. All conduits so far. Nothing has really happened to the air so much as we go from the atmosphere to these terminal bronchioles. You haven't taken out oxygen or added CO2 yet, water vapor, a few things like that, but you haven't had an exchange between the air and the blood at this point. It's all been conduits. So once we get to the terminal bronchioles, the next step we'll move into is actually the respiratory portion of the lung, where you get some exchange. Probably wait for that for next time. A couple more things with these airways I'd like to mention. Their histology changes, and not only the size. Of course, the trachea is a big honking tube with a huge cross-sectional area. As you branch, it gets smaller and smaller. The diameter, of course, gets much smaller. But if you went and took sections through the trachea, primary, secondary, tertiary bronchioles, down to the bronchioles, you would notice a gradual change in the histology, the makeup of these tubes, as you go from the biggest, the trachea, to the smallest. So I'm going to run a quick list down of what you've noticed. First of all, trachea, as I've already mentioned, it's primarily connective tissue, a lot of big C-shaped hyaline cartilage rings. As you move down the trachea, you'll notice the amount of cartilage is reduced. It gets into smaller plates in the bronchi, and by the time you get to bronchioles, the cartilage is gone. Bronchioles have no cartilage in them. Now, that's a potential problem because just like the trachea, the pressure inside these bronchioles is also lower than ambient pressure. Why don't they collapse? The reason they do not? Yeah, the trachea has a cartilage, the bronchioles do not. Why don't they collapse? Well, bronchioles are, of course, in the lung tissue. Now, if you look at what lung tissue is made out of, there's not much to it. It's not very massive. But it's basically epithelia, all those squamous epithelial cells making up these alveoli, these little air sacs we'll look at shortly. And also, it's got this huge framework of elastic fibers, elastic protein, which is stretchy. The lung can be modeled like a balloon. I mean, literally, if you take the lung and trachea out of something and you go, it'll blow up like a balloon. And if you stop breathing in it, it'll deflate. It's very elastic. You can model a lung just like a really thin-walled elastic balloon. It has a lot of what they call compliance, very high compliance. Small pressure changes, it inflates, changes its volume tremendously. So the bronchioles are embedded in this lung tissue. So if I kind of pretend to draw a picture of one, let's say this is a bronchiole right here. Let's make it this little circle. It's in the lung tissue itself, which is a bunch of elastic fibers. So imagine these little squiggly lines represent elastic fibers. It's almost like you've sewn them into the edge of this tube. All these elastic fibers are pulling. This is the force that will offset collapse. Even though the pressure change will tend to try and make this tube collapse, again, if it collapses, there'd be too much resistance, you couldn't breathe. The elastic fibers offset that. They kind of pull the walls of those smaller bronchioles open. So the term is radial traction. So all these elastic fibers pulling on the tube prevent it from collapsing. It doesn't need the cartilage like the trachea, which is just hanging out there, not supported by much of anything. Now you can see this if you have get my eraser. If you have, let's say, emphysema. Emphysema is kind of a chronic, progressive destruction of lung tissue. Imagine, this part of the lung tissue is destroyed. As it's destroyed, these elastic fibers also disappear. Now you're going to lose that support. Now, with this part of the lung tissue destroyed, if you try and inhale, the negative pressure will make this airway collapse. That's why in certain stages of emphysema, they can inflate the lungs, but when they try and inflate their lungs, all the airways collapse and they kind of breathe like it. Takes a tremendous amount of time for them to move air out of the lungs because the resistance in those airways has gotten too high. Again, air moving through tubes is just like blood moving through blood vessels. All the flow, pressure, resistance equations are basically the same. Air's a lot less viscous, but it follows the same rules. It's a fluid. So as you go from the trachea to the smaller bronchioles, the amount of cartilage is reduced and eventually disappears. Another thing you'll notice, the epithelia changes. Trachea, columnar, epithelia, ciliated. By the time you get to the functional portions of the lungs, that epithelia has changed to squamous epithelia. Why? Distance. Remember, when you want diffusion to occur, you don't want real thick membranes. Columnar epithelia is big fat stuff. Squamous epithelia is really, really thin. So you'll see notices or changes in the epithelia as well. And finally, muscle. There's not a significant amount of muscle in the trachea. There's some in the back. The back of these C-shaped rings have a little bit of muscle, but not much in the walls. When you get to the smaller bronchioles, there's a lot of smooth muscle in the walls of the bronchioles. Why is that? Well, why is there smooth muscle in your blood vessels? If the smooth muscle constricts, vasoconstriction, less fluid moves through it, there's more resistance. If the smooth muscle relaxes, vasodilation, there's less resistance, more fluid move through it. It's the same with the lungs. You can control the diameter of these smaller airways by relaxing or constricting the muscle. That's going to allow you to move to an area more easily or have more difficulty getting that area. It's to change resistance. Now, why would you want to do that? Partly it's for what they call ventilation perfusion coupling or ventilation perfusion matching. If you think about it, your lungs hold about 6 liters of air. But when you're breathing right now, you're sitting there at rest on the couch or wherever you're sitting, you're not moving 6 liters of air now. You're moving maybe about half a liter. But now think about it. You got this bag-like lung with a tube going into it, bronchi. Your whole body's basically a big bucket of water. You're about 80% water. When you inhale air passively, there were no way to direct it. Do you think the air is going to go to the top of the lung or the bottom of the lung? Well, most of the fluid's very dense. It's going to go to the bottom, blood, that sort of thing. The air is going to go to the top. So without any controls, you'd actually ventilate the top of your lung. You wouldn't ventilate the bottom much. But then think about the blood. Where's the blood going? Well, blood sort of has an option. It could go up here, or the middle, or the bottom. Without any controls, where's blood going to go? Well, gravity's going to pull it down to the bottom. So here is a potential problem you'd have. You ventilate the top of your lung, you perfuse the bottom of the lung, that is, send blood to it. How much oxygen you pick up? None. The air is going to a different spot than the blood. You wouldn't get any oxygen into the blood. It wouldn't work. So what we can do then, we know those blood vessels, these arterioles going into the pulmonary capillaries, they have smooth muscle in them that can contract or relax. And the airways have smooth muscle in them that can contract and relax. So what happens, an area of the lung that is not perfused, not receiving blood, the airways will constrict. So it doesn't receive air. That's going to direct the air to go to areas in the lung that are being perfused. So that's why it's ventilation, perfusion, matching. All it means is to be efficient, you want to send the air to the same spot in the lung where you're sending the blood. So by using a smooth muscle in the airways and the blood vessels, you can match those two. Where you're getting a lot of blood, dilate the airways. Where you're not getting a lot of blood, constrict them. That's going to send the air where you send the blood. So that's why you have changes in the smooth muscle. I'll go over some diagrams in a bit, real pictures, and go over these airways again so you kind of see what they look like. They don't look like this. So we'll call that part quits for the moment. Be back in a bit. Okay, here's a diagrammatic representation of those airways we just went over. So again, here's the trachea. It's a little prettier than mine. You can see those C-shaped hyaline cartilage rings. They give it a lot of support so it doesn't collapse. The larynx is up here. This is a cross section, so that represents that C-shaped hyaline cartilage ring.
And I didn't mention in the previous lecture, but why is it C-shaped and not a complete circle? It's open in the back here. That's because what's back here is the esophagus. Now, of course, when you're swallowing, what's going through your trachea? Hopefully nothing. So the esophagus, which is a muscular tube, can actually expand into the lumen of the trachea a bit so you can swallow larger items. And this is a diagram, again, of the trachea longitudinal section showing those pseudo-stratified columnar epithelial cells with the cilia, goblet cells. So here's that layer of mucus, and the cilia move it up, the mucociliary escalator, so you can get rid of the dirt and get the debris out of your lungs. This is showing you the airways. Again, here is the primary bronchi here. And then the secondary or lobar, you see you got 1, 2, 3 on the right, and there's just 1, 2 on the left. And then you see tertiary bronchi here. So that's the kind of separation. Now let me go one more. This is a cast of a part of the lower respiratory system. This is a trachea. They just poured some, probably, I don't know if it's a latex, a polymer of some sort down the airways and digested the organic material. So you're just seeing the airways. But that's a trachea. See the 2 primary bronchi, secondary bronchi, and then tertiaries, and all these are bronchioles. So that's why they call it a bronchial tree. Kind of looks like a dead upside down tree. But that is the bronchial tree. And let's see, I think that will do it. All right, one last bit here. This is showing you the kind of the bronchial tree and the trachea with the lungs intact. So here's a trachea, primary bronchi, 1 to each lung. This one's kind of showing you the lobes. That's that fissure there that separates the right lung into superior, middle, and inferior lobes. And here's your left lung. It's got a little inferior lobe here, and in the superior lobe, there's only 2. That's that cardiac impression there. That's where the left ventricle would project into the left side of the chest. And one more. Oops, here's where we go. This is where we ended. This is basically the terminal bronchiole right here. So this is the end of that bronchial tree. And what we'll get into next time, these terminal bronchioles lead into the respiratory portion of the lung. All these little clusters, sometimes described as little clusters of grapes, these are the alveoli. And they're showing you part of the pulmonary blood supply as well here. Pulmonary veins, pulmonary capillaries, pulmonary artery, pulmonary capillaries, pulmonary veins going back. These are the capillary networks around the alveoli, very dense. So we'll talk about the alveoli and gas exchange next time. Take care.